Good morning everyone. My name is Damodar. Just few days back I started my YouTube channel in order to share my knowledge on botany subject. So today let us discuss about a topic called as bacteria. Okay, so first of all, study of bacteria. Study of bacteria is called as bacteriology. Study of bacteria is called as bacteriology. So let us discuss about the history behind the discovery of bacteria. Anthony van Leeuwenhoek. Okay, Anthony van Leeuwenhoek is the first scientist who discovered bacteria. Okay, Anthony van Leeuwenhoek first discovered bacteria with the help of simple microscope discovered by himself. Okay, Anthony van Leeuwenhoek first discovered bacteria with the help of simple microscope discovered by himself and named them as animalcules. Okay, and named them as animalcules. Anthony van Leeuwenhoek first discovered the bacteria with the help of simple microscope discovered by himself and named them as animalcules. Okay, later on Ehrenberg, okay, first given the term bacteria. Okay, first given the term bacteria. So Anthony van Leeuwenhoek first discovered the bacteria with the help of simple microscope discovered by himself and named them as animalcules. Ehrenberg first given the term bacteria. Okay, so here Anthony van Leeuwenhoek first discovered the bacteria, hence he is called as father of bacteriology or father of microbiology. Who? Anthony van Leeuwenhoek. Okay, Anthony van Leeuwenhoek is called as father of bacteriology, father of microbiology. Okay, next Robert Koch. Next scientist is Robert Koch. Okay, Robert Koch named scientist first cultured bacteria. Hence he is called as father of microbial technique. Okay, first cultured, Robert Koch first cultured bacteria, hence he is called as father of microbial technique. Okay, so another important scientist named Louis Pasteur. Okay, Louis Pasteur, named scientist. So he given the concept called as pasteurization. Okay, pasteurization, where in this technique where harmful bacteria are removed. Okay, so here how this pasteurization is done. This pasteurization is done by HTTT, that means high temperature for low time. Okay, so here what they will do, near about 72 degrees Celsius temperature, nearly 72 degrees Celsius temperature is given for about 15 to 30 seconds. Okay, for about 15 to 30 seconds. Later on, LT, that means low temperature for long time. So near about 62 degrees or 61.5 degrees Celsius temperature for 30 minutes. So like this pasteurization technique is done, so that by pasteurization technique we can prevent the spoilage of food. Okay, where harmful bacteria are removed and the food cannot be, or the food can be stored for some hours or some days. Okay, so that is pasteurization technique. Apart from this pasteurization technique, so here L, if you take L for the Louis Pasteur first discovered liquid medium. Okay, first discovered liquid medium. Okay, and another important point is Louis Pasteur is the scientist who discovered germ theory of diseases. Louis Pasteur discovered germ theory of diseases. Okay, so where this theory states that disease to a living organism is caused by the bacteria. Germ theory of diseases. Because of these inventions, pasteurization and germ theory of diseases, Louis Pasteur is called as modern father of microbiology. Modern father of microbiology. Okay, modern father of microbiology. Louis Pasteur is called as modern father of microbiology. Okay, so here if you observe, still Louis Pasteur has discovered several other things. They are Louis Pasteur first given fermentation principle. First given fermentation principle. He discovered pasteurization. He has given germ theory of diseases. First given fermentation principle. Apart from this, a short code through which we can remember, that is CAR. Okay, CAR. Here CAR means, CAR code is used to study well about the vaccines discovered by Louis Pasteur. Here C for cholera, A for anthrax, R for rabies. Okay, so these are the inventions, or these are the important things that was discovered by the Louis Pasteur. Hence he is called as modern father of microbiology. Okay, he is called as modern father of microbiology. And one more scientist named Joseph Lister, okay, who discovered sterilization, like pasteurization. Here Joseph Lister discovered sterilization, where complete removal of microorganisms. This we follow in laboratory conditions in order to the apparatus like conical flask or petri dishes, what we use in the laboratory. So in order to remove the microorganisms, on those apparatus, we will use this sterilization technique, where complete removal of microorganisms occurs. Okay, so this is called as sterilization. Here in the pasteurization, only harmful bacteria are removed. Here complete removal, no microorganism will be there on the surface of that apparatus. So this is we follow in a laboratory condition, especially in tissue culture laboratories like that. So Joseph Lister is the scientist who discovered sterilization technique. Okay, so this is some history or the background that is about the discovery and the various inventions that was done by our scientists. So now let us see shapes of bacteria. So generally the bacteria may be circular or round. Circular or round. The circular or round bacteria is called as cocci. The circular or round shaped bacteria is called as cocci. So basing on this, these bacteria presents, cocci bacteria presents, how they are present, whether singular or multi or group. So basing on this, they are divided into following types. Monococcus, single circular bacteria. Single circular bacteria. Only single circular bacteria is present. If present, it is called as monococcus. Likewise, diplococcus. Two bacteria, two circular bacteria are present. If two circular bacteria are present, it is called as diplococcus. Next one, tetracoccus. That means here four bacteria are present. So those are called as tetracoccus bacteria. Okay, tetracoccus bacteria. Very important one in competitive exams, Sarcina. Okay, Sarcina, where bacteria are present 4 to 64 in cuboid fashion. Okay, Sarcina means 4 to 64 coccus bacteria are present in cuboid fashion. So that type is called as Sarcina. Okay, Sarcina, important. Next one is Streptococcus means a chain of cocci bacteria, like this, a chain of cocci bacteria. Okay, a chain of cocci bacteria. Next one, Staphylococcus means cocci bacteria present. Cocci bacteria present in the form of bunch of grapes. In the form of bunch of grapes. In the form of bunch of grapes. So such bacteria are called as Staphylococcus bacteria. So cocci means round or circular bacteria. These may be present in these following manner. Maybe monococcus. Monococcus, when we call it as a monococcus, when the single circular bacteria is present, it is called as monococcus. If two circular bacteria are present, diplococcus. 4 tetracoccus. 4 to 64 in the form of a cuboid, it is called as Sarcina. Streptococcus, a chain of cocci bacteria. Staphylococcus, cocci bacteria present in the form of bunch of grapes, like a bunch of grapes. It is called as Staphylococcus. Another shape is, this is the first one. Another shape is rod shape. Rod shape is called as bacillus. Rod shape of bacteria are called as bacillus bacteria. Okay, so this is also again classified as following types: monobacillus. That means single rod shaped bacteria. Monobacillus, single rod shaped bacteria. Diplobacillus, two rod shaped bacteria is called as Diplobacillus. Streptobacillus, that means a chain of bacillus bacteria. Okay, chain of bacillus bacteria is called as Streptobacillus. Palisade bacillus, that means here bacillus bacteria present in the form of stalks. Bacillus bacteria present in the form of stalks. Example, Corynebacterium. In the form of stalks, like a pile of stalks. Okay, that is called as palisade bacteria. Okay, so round is called as cocci, rod shaped is called as bacillus bacteria. Okay, next. Next shape is third one. Spirillum. Third shape is spirillum. That means so here shape is spiral shape. The bacteria shape is, if the bacteria shape is spiral shape, it is called as spirillum. Best example is Treponema palladium, which causes STD disease, sexually transmitted disease, syphilis. Okay, syphilis, STD disease called as syphilis. Okay, syphilis disease. Okay, it is spirillum shape. That means spiral shape. Fourth one is comma shape. Example is Vibrio cholera. Okay, Vibrio cholera, comma shape. The bacteria is comma shape. And fifth one Spirochetes. Spirochete. Here spirochete means spiral shape. Okay. Long, slender, spiral shape, spirally looks like corkscrew shape. Long, slender, spirally looks like a corkscrew shape. It is called as spirochete. This is also important. Spirochete. Here spirillum, important. Okay. So long, slender, spirally looks like a corkscrew shape. It is called as spirochete. Next one. Pleomorphic. Pleomorphic means bacteria changes its shape according to the environmental conditions. So pleomorphic bacteria changes its shape according to the environmental conditions. So that is called as pleomorphic bacteria. That means keeps on changes its shape according to the environmental conditions. That is called as pleomorphic bacteria. Okay. So next, these are some of the types of bacteria that may be classified based on the shapes of bacteria. These are some of the bacteria that may be classified basing on the shapes of bacteria. Okay. Next. Next is classification of bacteria. Based on flagella. Okay, based on flagella. Classification of bacteria based on flagella. Okay, flagella are the structures. Okay, flagella are the structures that helps in locomotion. That helps in locomotion. Flagella mainly helps in locomotion of bacteria. Okay, basing on these flagella, how these bacteria are classified, how many types these bacteria classified, let us see. Okay, so if flagella is absent, so bacteria in which flagella is absent, that type is called as Atrichus. Okay, that type is called as Atrichus bacteria. So that means flagella are absent. So flagella are absent. So those are called as Atrichus bacteria. If one flagella is present. If one flagella is present, so it is called as monotrichous. Those are called as monotrichous bacteria. Okay, only one flagella is present, so such type of bacteria are called as monotrichous bacteria. Next one is two flagella, one at each end. So monotrichous means here like this. So two flagella, one end each end, are called as amphitrichous. That means here, suppose this is the bacteria. On one end it is flagella is present, on another end another flagella is present. Two flagella, one at each end, are called as amphitrichous. Next one is two or more or tuft of flagella present at one end. Two or more or tuft of flagella, present at one end is called as Lophotrichous. Suppose this is the bacteria here. So tuft of or two or more flagella present at one end of the bacteria is called as Lophotrichous bacteria. If same two or more or tuft of flagella present at both ends, two or more flagella at both ends, cephalotrichous. Two or more or tuft of flagella present at both ends of the bacteria. It is called as cephalotrichous. Next, flagella. This is one. Next, flagella present all over the body. Flagella present all over the body of bacteria. Then it is called as peritrichous. Then the type is called as peritrichous. Okay, peritrichous. Atrichous means flagella are completely absent. Monotrichous means one flagella is present. Amphitrichous means two flagella, one at each end. So this is called as amphitrichous. Next here, two or more or tuft of flagella present at one end. So it is called as Lophotrichous. If two or more or tuft of flagella present at both ends, so then it is called as Cephalotrichous. Flagella present all over the bacteria body, then it is called as peritrichus. Then the type is called as peritrichus, and the shape and the bacteria looks like this. Peritrichus. So, basing on the flagella, the bacteria can be classified as these following types: atrichus, monotrichus, amphitrichus, lophotrichus, cephalotrichus, and peritrichus. Okay, next. Next, we will discuss about in detail structure of bacteria. In detail structure of bacteria. In detail structure of bacteria. So, if we observe the bacteria structure, one bacteria. So the inner membrane is called as plasma membrane. Next one is cell wall, and this is called as glycocalyx. Okay, glycocalyx. Together these three are called as cell envelope. They are called as cell envelope. Apart from this, so this is called as flagella. This is called as flagella, and they are called as pili, and short ones, these are called as fimbriae. So together pili, fimbriae and flagella are called as surface appendages. So here outer one, glycocalyx, cell wall and plasma membrane is called as cell envelope, and pili, fimbriae and flagella together called as surface appendages, and inner side this is called as nuclei. So are called as plasmid, inclusion bodies, ribosomes. Okay, so these are all together called as cytoplasmic inclusions. Cytoplasmic inclusions are contents. Cytoplasmic contents. So this is a typical structure of general structure of a bacteria. Okay, so let us discuss or let us see in detail about these structures. So first of all, we will discuss about cell envelope. Cell envelope, it consists of three structures. It consists of three structures. One is glycocalyx, cell wall, and the inner one cell membrane. Glycocalyx, cell wall, and a cell membrane. So glycocalyx, outermost layer. Okay, outermost layer is called as glycocalyx. This is classified as capsule and slime layer. This is classified as capsule and slime layer. Capsule is thick and strong, whereas slime layer is thin and strong or tough. Thin and weak. Okay, slime layer is thin and weak. Okay, outermost layer glycocalyx, outermost layer consists of, there may be capsule and a slime layer which is thick and thin. So the glycocalyx mainly helps in providing resistance to antibiotics. Mainly helps in providing resistance to antibiotics. Okay, next one is the cell wall. Next one is cell wall. Here glycocalyx is made up of mucopolysaccharides. Okay, glycocalyx is mainly made up of with mucopolysaccharides. Glycocalyx is mainly made up of mucopolysaccharides. Outermost layer comprises of capsule and slime layer. Capsule is thick and slime layer is thin. Mainly provides resistance to antibiotics. Made up of mucopolysaccharides. Okay, next cell wall. Cell wall is made up of peptidoglycans. Okay, which comprises peptides and peptides and carbohydrates, which comprises peptides and carbohydrates. Cell wall is made up of peptidoglycans, which comprise of peptides and carbohydrates. Here peptides consists of near about 4 to 5 amino acids. 4 to 5 amino
కలిపి పెడుతుంది ముద్దగా తనలాలి పాటలోని సరిగమ పంచుతుంది ప్రేమ మధురిమ పెదవే పలికిన మాటల్లోనే తీయని.
Four to five amino acids. Carbohydrates consists of NAM and NAG. NAM and NAG. Carbohydrates consists of NAM and NAG. Here NAM means N-acetyl muramic acid. NAG means N-acetyl glucosamine. Okay, so NAM and NAG carbohydrates are present. So made up of peptidoglycans, consists of peptides and carbohydrates, 4 to 5 amino acids, carbohydrates, NAM and NAG. And coming to the last one, cell membrane. Cell membrane. The cell membrane of bacteria is similar to eukaryotes. Is similar to eukaryotes. That means it is made up of, cell membrane is made up of lipids and proteins. Cell membrane is made up of lipids and proteins. Similar to eukaryotes, made up of lipids and proteins. Okay, this is about the cell envelope. Okay, this is about cell envelope, which consists of glycocalyx, cell wall and cell membrane.